molecular basis of inheritance. So before we start this chapter, let's see a few of the experiments that are really important. So the first one is the Griffith experiment. He's a scientist called Frederick Griffith. So he was working, at the time of the World War One or two. I think it's World War Two. So at the time, so many people were affected with the pneumonia. So this guy was researching over the bacteria which is causing the pneumonia, that is the Streptococcus pneumonia. Around in the year 1928, he was working on this Streptococcus pneumonia. So there he observed two strains of Streptococcus. One is the S strain, which is also called as the smooth strain, and the other one, it is a rough strain, which is a R strain. Okay, remember in that smooth strain, it is very virulent. That means it is very, very dangerous. Remember this. So this S strain is capable of causing the pneumonia so that the mice will die. Whereas the R strain, it is not so virulent. That means it's not very dangerous because this is lacking the protective capsule. Whereas here there is a protective capsule is present. That capsule is made up of polysaccharides. And because of that capsule, it is giving very smooth texture. Okay. Whereas here there is a lack of this polysaccharide capsule. Lack of protective capsule. Remember this. So because of this, it is rough strain. Okay. So see here, the bacteria Streptococcus pneumonia was grown on the culture medium, and he observed that two strains are there. One is S strain, the other one is R strain. And this S strain is having outer side a mucus polysaccharide coat, which is very, very dangerous. Whereas the R strain, it is not having such a coat. So it is not a virulent. Virulent means it is a very dangerous. Okay, remember this. Then he observed four experiments. Okay, so like first, what happened is he injected the S strain, which is live, into the mice, and he saw that the mice was died. So, and when he extracted the plasma, he observed that there is a S strain bacteria is present in that plasma. Understood? Then he did the live R strain, injected into the live mice. The mice is live. And when he extracted the plasma, there is no R strain because the mice immunity has killed the R strain. But here, the mice is died because of this live S strain. So S strain has killed the mice here. In this case, the mice itself killed the R strain. Understood? So what happened in the step one? So he injected the live S strain bacteria into the mice, and that is result in the pneumonia and death. So just remember the step one is injecting live S strain into the mice. So result in pneumonia and death. Whereas when he injected the live R strain into the mice, there is no ill effects. Remember this. So there's nothing happened. So injected the mice with live S strain, there is a virulent and the mice died. So he isolated S strain bacteria from the dead body. And he did this next step. He injected the mice with live R strain and non-virulent. So the mice is survived, but there is no living bacteria because the mice immunity has killed the R strain. Is this clear?
Yes, sir.
Yeah. And then he did the third experiment. In that, what he did is he injected the eat killed S strain bacteria. Okay. So when he just injected only the eat killed S strain, just take this. When he injected dead S strain, mice is alive. But when he injected this dead S strain with the live R strain, then the mice is died. Okay, so the mice has died, and he confirmed that. So there is something that has moved from the eat killed S strain to the live R strain, so that now this live R strain has changed to a very deadly combination. You understood this? So when you
Yes.
injected the mixture of eat killed S strain and the live R strain into the mice is dead. And he confirmed that there is something that has passed from the S strain to the R strain, so that the R strain has transformed into the S strain. Understood? Is this clear?
Yes. Yes, sir.
Pridila, what about you?
Yes, yes.
So what happened in this? Okay, so he observed in the fourth step. This is the third step. This is the fourth step. So in the fourth step, Griffith observed the mice was died because of the pneumonia, and he saw that there is a living strain here. Okay, so there's a living strain. But when he injected the dead strain. But when he extracted living strain. So from that, he confirmed that whatever the live R strain is there, this live R strain has changed to S strain. How it has changed? What is the difference between R strain and S strain? In the R strain, there is no outside polysaccharide coat, but the S strain is containing that outer polysaccharide coat. So that has created. And how does it created? There is something that is passed from the S strain to the live R strain, which has that information to create this polysaccharide coat. Understood? So this is the unexpected outcome that is seen in the Griffith experiment. Is that clear?
Yes, sir.
From this fourth step, the Griffith understood that there is something that allowed to the R strain to acquire the ability of this S strain. Okay, and what is that? We don't know. Okay, I mean at that time the people doesn't know what is that. So see here, when S strain and R strain bacteria are mixed, the non-virulent strain bacteria is transformed into the virulent S strain bacteria, and that process we call it as transformation. Okay, the S strain as the R strain has transformed into the S strain. And what is the transforming principle which made the R strain to the S strain? What is that information that is passed from the S strain to R strain so that this R strain is converted into S strain? That we call it as transforming principle. Understood what is meaning of transforming principle?
Understood. Yes.
And this has enabled the R strain to synthesize smooth polysaccharide coat and become virulent. Okay, due to the transfer of the genetic material. So whatever this transforming principle is there, that is nothing but the genetic material. But that genetic material is DNA, RNA, proteins, or carbohydrates. We don't know at the time. Okay, I mean they don't know at the time. Is that clear?
Yes. Yes.
Yeah. So see here, live R string, mouse dies, live R string, mouse lives, sorry, live S string, the mouse dies. Eat killed S string, mouse lives. But when you add the live R string with the eat killed S string, the mouse dies. So now the question is, what is this transforming principle? Is it a carbohydrates? Is it RNA? Is it a protein? Or is it a DNA? What is it? So we don't know what is the chemical nature of this transforming principle. So we need to find out which one is the transforming principle in this. Okay. The Griffith experiment was the turning point for the discovery of the genetic material. Okay. So from this, till that time, what they believed, there is a, till that time, what they believed,
That RNA was the genetic material.
one second, guys. Okay. Till the time, what they believed is whatever the genetic material is there, it is unchangeable. Okay. Whatever the bacteria types are there, those bacteria types are fixed and they can't be changed. Okay. So they are unchangeable. They are fixed. But after this, they understood that whatever the bacteria type is there, it can change if you pass out the information. And that passing out the information is the transforming principle. And what is the transforming principle? The chemical formula, I mean, the biochemical nature of this transforming principle, they doesn't know whether it's a carbohydrates or DNA or proteins or DNA. Okay. So remember this. However, it failed to explain the biochemistry of the genetic material. The biochemical nature of the genetic material was unknown. It could be proteins, carbohydrates, RNA, or DNA. Is this clear?
Yes.
Yeah. So Griffith work, it was inspired the other scientists like Avery McLeod, MC Carty. So they did the further investigation. They continued the Griffith experiment in the year 1944. If you see the Griffith experiment, it is almost done in the year 1928. But these people, they continued in the 1944. So till 1944, we don't know what is the genetic information. Is that DNA or RNA or what? We don't know. Okay. So every MacLeod and MC Carty, these people, they did the experiment and they identified the DNA is the transforming principle in the year 1944. Okay. With a solid, solid evidence. We'll see what is that solid evidence. For suppose what they did is they purified the biochemical nature of the carbohydrate, protein, DNA, and RNA. Okay, from the eat killed S string. What they did first, they took the S bacteria, S strain bacteria. After they took the S strain bacteria, they exposed it to the heat. After that, they took the proteins, they took the carbohydrates from this bacteria. They extracted these things, carbohydrates into one test tube, proteins into one test tube, and DNA into one test tube, RNA into one test tube. These are the major biochemical things that are present inside the cell. So they extracted all these biochemical things into different test tubes. So one is the carbohydrates, RNA, proteins, and DNA. Now what they did, so in each of these test tubes, they also added the bacteria, R strain bacteria they added. Okay. So remember, so to each of these solution, they added the live R strain bacteria. Understood? If my transforming principle is proteins, then this will show the R strain to S strain conversion. What is this? I don't have a. One second, guys. Okay, I need to send another link once this link is over because my premium subscription is over. I need to recharge. Sharon, do you have a physics class today?
Yes.
Okay. Okay, fine. I'll do that. So see here, if my genetic material is the proteins, okay, then I'll be seeing the transformation of the R strain to S strain in the blue test tube. If my uh, genetic information transforming principle is RNA, then I'll be seeing the R strain conversion to S strain in the green test tube. If it is sugar, then I'll see here. But if it is the DNA, then I'll be seeing in this. Understood? What I'm trying to say here. See, for now, you just remember the DNA is the transforming principle. We know that after finding out this experiment. But before this experiment, we don't know. So we'll be putting all the R strains into each of these test tubes and seeing where the conversion will happen. Where does the R strain to S strain will happen? Does it happen here? Does it happen here? Does it happen here? Or does it happen here? So I'll be checking out. And I found that so in sugars, in RNA, in proteins, it is not happened. It happened in the DNA test tube or the test tube containing DNA. But still, I want to confirm it. So what did I do? So see here, sugars, there's no transformation, RNA, no transformation, protein, no transformation, but DNA, you will be seeing the transformation. But I want to confirm it. So what do? Except this DNA, remaining all the test tubes, I'll put the RNAs, carbohydrates, protease, so that all the proteins, RNA, sugars will be digested. But only I keep the DNA intact. Still the transformation is shown. Then I will further continue it. Now what I do, I'll put all the test tubes with the DNAs. But I'll keep the sugar intact, RNA intact, protein intact. Okay, because I'm not digesting the proteins or RNA or sugar, but I'm only digesting the DNA if any test tube contains the DNA. Then for my thing, I understood that once I added the DNAs, there is no transformation at all in any test tube. So from that, I concluded that DNA is the pakka genetic material. Understood? So they
Yes.
showed this solid evidence that DNA could be the genetic material. Understood?
Yes. Understood.
Yeah. So you see here, to confirm that DNA is the transforming principle, they added the carbohydrates, RNAs, and the protease, so that all the carbohydrates, RNA, and the proteins will digest. So that still showed the transformation because I didn't add the DNA is. But in the next experiment, I also added this DNA S. There is no transformation. From that, they showed the evidence. It would be the DNA, the genetic information or the transforming information. Is that clear?
Yes, yes, sir.
Okay, fine. Is it showing any timer for you?
No, sir. No,
Okay.
sir.
Okay. So see here, they concluded that DNA is the hereditary material, but not all biologists were convinced. Okay. Still they think because DNA is just made up of adenine, guanine, cytosine, thymine. But how can these four repeating nucleotides can store that much genetic information is the question for many scientists. And on the other hand, protein, there are many amino acids, there are many proteins in the body. So proteins could be the genetic information because it is very diverse. On the other hand, this DNA is just made up of only four nucleotides. How can it store that much big information? Is the question mark for the many scientists. So see here, at the time, belief was DNA was just a chain of four repeating nucleotides and it was not complex enough to store the genetic material. On the other hand, protein was very diverse, so it could be the genetic information. That's the what other scientists used to argue. So, in order to prove, like without any exception, that we call it as unequal proof. This word is clearly mentioned in the NCRT also. The unequivocal proof is given by Hershey and Chase experiment. Remember this. Understood? By using the bacteria. Okay, I'll be sending you the the Zoom meet again. There's only 4 minutes left. I'll send, join the new meeting. Okay?
Yes, sir. Okay.
Yeah. See, according to some thesis, they say that RNA is the one which is first originated, from which the DNA has evolved. Okay. And RNA is not very good genetic material compared to DNA. So nature has selected this DNA as the fundamental molecule of heredity. That means it will carries the genetic information from one organism to the other organism. From one generation to other generation. Okay. So you see here, this is the double helix structure of this DNA. Okay. If you see here, there is a major groove and the minor groove. Okay. So you see here, there is a major groove and minor groove. And if you see this double helix, these double helix are joined by the all the nucleotides. There is a pair of nucleotide. So this is one pair, this is one pair of nucleotide. This is another. So what do we say? Pair of nucleotide. Okay. So pair of nucleotides. That means there are two nucleosides on the opposite strands. So if you see here, this is one strand and the other one is the another strand. So this is one strand and this is another strand, which are complementary to each other. So one nucleotide of one strand, another nucleotide of the another strand, they join together. So that's how they are making a pair of nucleotides. We'll be seeing in detail how these nucleotides are all joined up. Okay. And remember, the distance between each of the nucleotide is 0.34 nanometers. Understood? So what is the distance between two base pairs? It is 0.34 nanometers, or you can say 0.34 into 10 power minus 9 meters. Okay. Remember, this is very, very important for calculations. There are many calculations come on this thing. Okay. So they will be asking the length of this DNA. They will be giving certain number of nucleotides and they will ask what is the length of the DNA. We'll calculate the DNA length in the eukaryotes and prokaryotes. Please confirm my voice is audible, right? Is there any background noise?
It's audible, sir.
Okay. Okay. And one more thing you need to remember is plectonomic. Okay. So DNA is a plectonomic in nature. Have you heard this word? What is meaning of this plectonomic? See, DNA is a plectonomic in nature. That means if you see here, there are two strands are present. Yes or no? This is one strand and this is another strand. So these two strands, you cannot separate them until you completely unwind it. For suppose if I just open the strand like this, so this is the strand and I open the strand like this, but still I didn't unwind completely. That means still my DNA cannot be separated. So you completely unwind the both the strands, then only you can break the double bonds, you can break the hydrogen bonds, and you can separate the two strands. Till then, you cannot separate the DNA, the two strands of the DNA. So that's what the plectonomic. Okay. So understood what is meaning of DNA is plectonomic in nature?
Yes, sir.
Yeah. Just remember this. DNA is plectonomic in nature. That means two strands of DNA can't be separated until it is completely unwinded. Okay, cannot be separated without completely unwinding it. Banu, understood? What is this means? Okay, so see here, there are two strands are present. Each of them are anti-parallel in nature. That means if you see one of the strand, it will be 5 to 3, the other one will be 3 to 5. So both the strands are parallel to each other. That means they have a equal distance between the two opposite base pairs. So two opposite strands, the distance is always equal. Okay, but the polarity is opposite. You can see here one is 5 to 3, the other one is 3 to 5. So they are said to be anti-parallel polarity. So we say anti-parallel polarity. Okay, so one will be in the 3 to 5, the other one will be 5 to 3. And if you see here, this DNA is completely coiled. And this coil is happened in a right hand manner. Remember this. The coiling of the DNA through the common axis, it was happened in the right hand manner. That means you can say it's a clockwise direction it happened. So spirally coiled itself around the common axis, you can say in right hand fashion. Okay, so remember this. So it was spirally coiled around common axis in a right-handed fashion. Is that clear? Any doubts?
No, sir.
Okay. So know these things. One is DNA is plectonomic in nature. That means the two strands cannot be separated until you completely unwind them. Second, these are anti-parallel polarity. That means one is 5 to 3, the other one is 3 to 5. And it is right-handed manner. Whatever this spirally coil, it was happened in a right-handed fashion. And the other one is the distance between the two base pairs is 0.34 nanometers. Okay. And we call, for suppose if you see here, from year to year, there is a one complete turn. That means this was happened like this, 180 degrees, and there is like this, 180 degrees. So if you see here, it was happened 180 plus 180. That means one complete turn, 360 degrees. Okay. So this one complete turn, we call it as the pitch. What do we call? The pitch of the DNA. Okay, so what is meaning of pitch? It is nothing but one complete turn. Okay, so this one complete turn will contain 10 base pairs. Okay, remember this. The pitch of the DNA contains 10 base pairs. And what is the distance between two base pairs? Mridula?
0.34 nanometer.
Yeah, that means 10 into 0.34 nanometers. You can say 3.4 nanometers is the pitch of the DNA. That means from year to year, the distance is 0. Sorry, 3.4 nanometers. Understood? So just remember, pitch that you can say one complete turn, or you can say one complete helix turn, and it contains total 10 base pairs. So the 10 base pairs, the distance is 10 into 0.34 nanometers, that is 3.4 nanometers. Clear? Let's talk about this DNA. Okay, so this DNA is made up of nucleotides. So DNA is a polymer and it is made up of a single monomer that is called as a nucleotide. So this nucleotide is consists of three things. One is a 5 carbon monosaccharide. Second, it consists of nitrogen base. This nitrogen base, it is heterocyclic. I'll tell you what is the heterocyclic. So it is heterocyclic in nature. Okay. Next, it is containing the phosphoric acid, or you can say phosphate ion. If it is present in the form of ion states, we call it as phosphate ion. When it is present in the form of anion, or when it is present in the neutral charge, we call it as phosphoric acid. Remember, so it is containing three things. One is 5 carbon monosaccharide, or you can also call it as the ribose sugar, deoxyribose sugar if it's a DNA. If it's RNA, we call it as ribose sugar. And the nitrogen base, which is heterocyclic in nature. I'll tell you what is this heterocyclic. And it is containing the phosphate ion. So these are the three things it is made up of. Okay, so remember this: 5 carbon sugar, nitrogen base, and phosphate ion or phosphate group. So let's see more about this pentose sugar. If you see here, this pentose sugar means it is made up of 5 carbon. Okay, so this is the first carbon, second, third carbon, fourth carbon, and the fifth carbon. Okay, so let's focus on the second and third carbon. Okay, always the third carbon contains the OH group. If it's in the DNA or RNA, it contains the OH. Okay, you can see here OH, OH. But if it contains the OH also on the second carbon, we call it as the ribose sugar. You see here, on the second carbon, there is OH is present. But if you remove the oxygen, then only the hydrogen will be present. Then you call it as deoxyribose. As we are removing the oxygen, so we call it as deoxy. Remember this. Okay, so remove oxygen on second carbon. Okay, we call it as deoxy, which is nothing but a DNA. DNA is made up of deoxyribose sugar, whereas RNA, it contains ribose sugar, where on the second carbon OH is present. On the first carbon, you will be having the OH. On the fifth carbon, you will be having the OH group, which will be helpful for attaching to the nitrogen base and the phosphate group. We'll see that. Okay, but remember, this is the important thing. On the second carbon, if it is hydrogen, we call it as deoxyribose. On the second carbon, if it is OH group, hydroxyl group, we call it as ribose sugar. Okay, so that's the difference between the DNA and RNA. First main difference. On the second carbon, if you have OH, it will be RNA. On the second carbon, if you have hydrogen in a nucleotide, we call it as DNA, deoxyribose. Is this clear?
Yes, sir.
So pentose sugar, as there are 5 carbons are involved. So let's see, next one is the bases. Bases are two types. One is the purines, the other one is pyrimidines. One is purines. Okay, so you can say this adenine and guanine will comes under purines. So what is purines? Purines are nothing but they are two rings. They are made up of two rings. You can see here, this is one ring, this is the second ring. This is a one ring, this is the second ring. So it is made up of two rings. One is 5-membered ring. The other one is 6-membered ring. Remember this. Okay. So you see here, this is 1, 2, 3, 4, 5, 6. So 6 member. If you see here, this one, 2, 3, 4, and 5. So one is 5 membered, the other one is 6 membered. Both fused and result in formation of purine. If you see here, this 5 membered is fused with the 6 membered. So this 6 membered is also called as the pyrimidine. Remember. Okay. And the 5 membered ring, it is called as imidazole ring. You will be studying in the biomolecules, but now you just remember the 5 membered, it is a imidazole. The 6 membered, we call it as the pyrimidine. Okay, both fuse it together and result in formation of a purine. So by this you can say purines is nothing but 5 membered ring plus pyrimidine. Okay, so just remember purines is nothing but 5 membered ring, or you can say pyrimidine plus imidazole, or you can say pyrimidine plus 5 carbon ring, not 5 carbon, 5 membered ring. Okay. Yeah, we said the bases are heterocyclic. Why we say heterocyclic is, if you see here, all the 6 membered or 5 membered, they are not maintained by the carbons. There is a nitrogen and there is a carbon, nitrogen, carbon, nitrogen, carbon. That means different groups were present within this ring. So we call it as heterocyclic. If all are carbons, then we call it as homocyclic. But this ring is made up of different compounds. For suppose this junction is having nitrogen, this junction carbon, this junction nitrogen, this junction carbon, this junction carbon, this junction carbon. So it's made up of mix of nitrogen and carbon. So we call it as the heterocyclic ring. Is that clear? So why do we call the nitrogen base as heterocyclic? Come on, guys. Am I audible?
Yes, sir.
Mridula,
Yeah, sir.
what happened? You're not talking. Banu, are you there?
Yes, sir.
You understood this?
Yeah, sir.
Okay. So remember. And what comes under these purines? It is adenine and guanine. Remember this. This is also very important thing. So what comes under this? A and G comes under purines. Okay. Is that clear? Any doubts so far?
No doubt, sir.
Okay, perfect. So next one, pyrimidines. Pyrimidines are just a normal 6 membered ring. Okay. So 6 membered ring, it will be helpful for forming the purines as well. So what comes under pyrimidine is cytosine, thymine, uracil. And remember, always a purine binds with pyrimidine. For suppose you see here, adenine. Adenine binds with thymine or uracil in RNA. Guanine binds with cytosine. So if you see A binds with T, or A binds with U, or G binds with C. Okay. If you see A and G, these are purines. But if you see T, U, C, these are pyrimidines. So always a purine bound with a pyrimidine. So that's how they are maintaining equal distance between them. So whatever the distance between them, always that it is equal between the purine and pyrimidine. So they are parallel in nature. Remember this. And remember, adenine and thymine, guanine and cytosine, you will be seeing in the DNA. Adenine and uracil, guanine and cytosine, you will be seeing in the RNA. Remember this. Because in DNA, you will be having thymine. In the RNA, instead of thymine, uracil is present. This is the second thing. What is the first difference? The first difference is on the second carbon, if you have hydrogen, we call it as ribose sugar, that's RNA. On the second carbon, if you have OH, it is deoxyribose sugar, which is a DNA. So that's the first difference between the RNA and DNA. The second difference is if the thymine is there, it is a DNA. If uracil is there, it is a RNA. Clear? The two
Yes.
major differences between the DNA and RNA. So you can see the structures are also important. So just remember them. It will be helpful for in the biomolecules, you will be studying these structures. So next one. So this nitrogen base, if it joins with a sugar, okay. So remember, adenine, guanine, cytosine, uracil, thymine, these are not nucleotides. These are nitrogen bases. Okay. If this nitrogen base joins with the sugar, it could be ribose sugar or deoxyribose sugar. Okay. So remember, if this nitrogen base, it joins with the sugar, we call it as nucleoside plus sugar. Okay. It could be ribose sugar or deoxyribose sugar. Ribose sugar we'll simply call adenosine. If it is deoxyribose sugar, we mention the word deoxy. Okay. So you can write like this. Adenine, when it binds with a ribose sugar, we call it as adenosine. The words are very, very important. Please remember them. It is a nucleoside, not even a nucleotide. Remember, this is a side. Okay. So see here, first the nitrogen base. The nitrogen base, when it joins with the sugar, it will make the nucleoside. Nucleoside. This nucleoside, when it joins with the phosphate group or phosphate ion or phosphoric acid, it will make the nucleotide. Remember this. Okay. So nucleoside means it's only nitrogen base and sugar. Nucleotide means nucleoside plus phosphate group. That will make nucleotide. So now we are talking about the nitrogen base, and when it joins with the sugar, what is the name of this nucleoside? Okay, still it's not a name of the nucleotide. Remember, that's very important. Why I'm stressing is sometimes they will give what is the name of this nucleotide, and they will mention some adenosine. You will be wrong if you mention the adenosine, because that's not a nucleotide, it's a nucleoside. Or they will say which nucleoside is formed. If you write adenine, it is wrong. You should not write adenine, you should write adenosine. So the word spelling is very, very important. So see here, adenine, when it joins with the sugar, so it will become a nucleoside. We call it as adenosine. This is very important. Adenosine. If it is a RNA, okay. If it's a DNA, then you can call it as deoxy. Remember this. Okay. So adenosine or deoxyadenosine. Guanine, it will become guanosine. SING. If it is cytosine, see here again, you should not mistake it like a nucleoside, as it is NOSING. Okay. Cytosine, it will be called as cytidine. See here, cytosine is not a nucleoside. It is just a nitrogen base, even though it ends with the word cytosine, SING, but still it's a nitrogen base. Uracil, uridine. Thymine, thymidine. So remember these nucleoside names. So adenine will give adenosine or deoxyadenosine. Guanine will give guanosine. Cytosine will give cytidine. Uracil will give uridine. Thymine will give thymidine. Clear? Any doubts?
No, sir.
Okay. So see here, how do we join these? Okay. So further, now this cytidine or these nucleosides, now they will be joining with the phosphoric acid. They will be joining with the phosphate group. So it will give nucleotide. Remember this. So how does this will be joined? Okay. First, suppose you can see here. Okay. On the fifth carbon, the phosphate group is attached. Okay. On the first carbon, the nitrogen base is attached. Okay. So this is the phosphate group, this is the nitrogen base, and this is a sugar. So we called it as the nucleotide. Okay. At this nucleotide, on the second carbon, you have OH. Sorry, on the third carbon, you have OH. If you have OH on the second carbon, you call it as ribose sugar. If you have hydrogen on the second carbon, you will call it as ribose sugar. That's a nucleotide. Okay. Or deoxynucleotide. That depends whether it's a RNA or DNA. So now concentrate here on the third carbon, you have OH. And if you see this phosphate group, actually this phosphate group is like this: P double bond O, P OH, OH, and OH. So this is called the phosphoric acid. When it loses the hydrogen, this will turn out to be PO minus, PO minus, and PO minus, which is a phosphate ion. Actually, the negative charge in the DNA is because of the phosphate group. That phosphate group is containing the negative charge. So it is making the entire DNA negative charge. That one thing you remember. On the on this phosphate group, there's a OHR present. These OH will be helpful for joining this phosphate group with the other phosphate, as well as with the other nucleotide, as well as with the nucleotide. For suppose, let's see how it will do. First one, let's take one, let's take two, and let's take three. Okay. So first one, this is the fifth carbon. If you see here, this is the pentose sugar, and there is a fifth carbon. This fifth carbon, it is having OH group, and the phosphate is also having OH group. So this hydrogen and this OH, they will join up together and remove like a water molecule. So now this carbon will join with the oxygen. So this bond, we call it as phosphoester bond. Understood? So this is called the phosphoester bond. How it is formed? By removing the water molecule. So that's how the OH group on the phosphate will be helpful for binding to the carbon, the fifth carbon. Second, it will be helpful for binding to the other phosphate group. If you see the other phosphate group, it will be like this: P double bond O, OH, or let's say like this: P double bond O, OH, OH, OH. Let's say this is attached to the fifth carbon. Okay. Take one more phosphate group, which is having OH, OH, and OH. Now see here, there is a hydrogen is present, there is a OH is present. They join together and it will be helpful for attaching one phosphate group with the other phosphate group by removing the water molecule. Okay. Now here you remove the water molecule, but this is not a phosphoester bond. It is an anhydrous bond, because here you join two molecules by removing the water molecule. That kind of bond, we call it as anhydrous bond. Okay. So that's how it become a diphosphate group. And if you attach on the left corner another phosphate group like this, then it will become triphosphate. So that's how we call ATP, ADP, AMP, based on how many phosphate groups are present. And remember, this anhydrous bond is formed, which is a very, very strong bond, and breaking this bond will liberate a lot of energy. So that's how you remove the bond here by breaking the ATP to ATP. You convert the ATP to ADP plus inorganic phosphate. It will leave a lump of energy as you broke this bond. You break the bond here again. So ADP will convert to AMP plus inorganic phosphate. This will again release a lump of energy. So that's how a triphosphate stores the energy in the form of the bond, which will be helpful for cellular energy requirements. So that's why it is called as the cellular currency, ATP. As you break the phosphate bond on it, it will leave lump of energy. As you break the diphosphate to monophosphate, it will leave the lump of energy. Is that clear? How the second phosphate or the OH group on the second phosphate will be helpful for attachment to the other phosphate groups?
Yes, sir.
Okay. So this is how the attachment was happened between the two phosphate groups or three phosphate groups. One attachment is helpful for making bond the phosphate with the fifth carbon. Second one will be helpful for joining the three phosphates. Now the third OH group, it will be helpful for suppose, let's say, see, double bond I am taking here. Let me take the single bond here. Okay, let's take this complete white bond like single bond. Okay. So just take this as a complete single bond. Don't take it like a double bond. So it is having OH. And if you see here, there is a OH on the third carbon. There is a OH on the phosphate group. Now they will be joining again and releasing the water molecule. Okay. Now it will establish the connection between the phosphate and the third carbon. So that's how it will be helpful for one nucleotide to bind with the other nucleotide. Okay. And if you see here, there is a phosphoester and there is a phosphoester. Between these two phosphoesters, the bond has created. So this bond, we call it as phosphodiester bond. Is that clear? How does the phosphodiester bond has formed? Or you can say this as a phosphodiester linkage. Okay, so the phosphodiester linkage will happen between the fifth carbon and the third carbon. Okay, of the pentose sugar. Okay, fifth carbon. Because if you see here, this phosphate group is present on the fifth carbon. So it will be happening between the phosphate group and the third carbon OH group. So they will be making the bond. That bond we call it as phosphodiester linkage. So that's why this bond direction is also said to be 5 to 3. Because the fifth carbon phosphate and the third carbon OH, they will be making this phosphodiester bond. So it is between 5 and 3. Is that clear?
Yes, sir.
Any doubts so far? So remember this one thing. So that's how two nucleotides were joined by using this phosphodiester linkage. So just remember this. Nucleotides joined together, okay, by phosphodiester linkage. Or you can say phosphodiester bond. Now this phosphodiester linkage was happened between the fifth carbon and the third carbon. Or you can say the phosphate group on the fifth carbon and the hydroxyl group on the third carbon. Clear? Shrikar, అర్థమైందా?
Yes, sir.
What about you, Banu? Ridula, you understood, right?
Yes, sir. Understood.
Okay. So you can see here. Okay. So this is the glycosidic bond, or you can say N-glycosidic linkage. This is said to be N-glycosidic linkage. That means the bond happening between the nitrogen base and the pentose sugar. So we call it as N-glycosidic bond or N-glycosidic linkage. And between the fifth carbon and the phosphate group, we call this bond as phosphoester bond. Okay. And between one phosphate group and the other phosphate group, we call this bond as anhydrous bond. And between one nucleotide to the other nucleotide, we call it as, I'll show you in the next slide. Okay. We call it as phosphodiester bond. Remember this. So there are total 5 bonds are present. One is the anhydrous bond, which is present between triphosphates. One is the phosphoester bond that will be helpful for attachment of phosphate to the fifth carbon. One is the N-glycosidic bond that is helpful for attachment of nitrogen base on the first carbon. The other one is phosphodiester bond, which will be helpful for attachment of one nucleotide with the other nucleotide. Okay. And there is a hydrogen bond is also present, which will be helpful for attaching one nitrogen base with the other nitrogen base in the opposite strands. So you can see here. So this is the phosphate group, this is the nitrogen base, and this is the pentose sugar. So which will make completely a nucleotide. And these nucleotides, polymer, we call it as nucleic acid. It could be the DNA or it could be the RNA. If it is RNA, it will be having OH. If it is DNA, it is having H. So OH if it is RNA, H if it is a DNA. Okay. So see here, one nucleotide attach it to the opposite strand nucleotide with a hydrogen bond. And if you see here, between the thymine and adenine, there is only two bonds were present. Okay, so just remember adenine and thymine, they are bound with the two bonds between them. If you see the guanine and cytosine, there are three bonds were present. 1, 2, and 3. So guanine and cytosine, three bonds were present. Okay, and remember adenine and guanine, they are purines. Thymine and cytosine, they are pyrimidines. And the adenine also binds with uracil if it is RNA. This is DNA. This is both DNA and RNA. Okay. Okay. So you will be seeing the adenine and thymine exclusively in DNA, adenine and uracil exclusively in RNA, guanine and cytosine both in DNA and RNA. Okay. So remember this. So there is always a pyrimidine makes double bond or triple bond with a purine. Okay. So you can write the purine makes a double bond or triple bond with the pyrimidine. So here adenine and guanine, they comes under purines. Okay. So on my left, I have purines. On my right, I have pyrimidines. So they are bound either by double bond or triple bond. And remember, always adenine pairs with thymine and guanine pairs with cytosine. And adenine in RNA pairs only with uracil. Okay. And the bonds present between them is a hydrogen bond. Is that clear? Any
Yes.
doubts? You can see here, this is how the sugar and nitrogen base between them. See here, always the base will be inside and the sugars and phosphate groups will be outside. So sugars and phosphate group is outside, bases will be at the center. So that's how it will make. Sugar and phosphate is outside, bases are at the interior position. And remember a few things about the anti-parallel polarity. So DNA, it contains the two strands, whereas RNA contains only one strand. Okay, and here two main strands, they have this phosphate backbone. Remember this. So they have a phosphate backbone. And normally, if you see the joining happen between the fifth carbon and the third carbon of the other nucleotide. Okay, so remember, joining of two nucleotides is happened between the fifth carbon phosphate group and third carbon hydroxyl group, third carbon OH group. Okay, so see here, fifth carbon phosphate group and third carbon OH group, they are making a bond. That bond name we call it as the phosphodiester bond. This is the second thing you need to remember. And the polarity of the two strands, one will be 5 to 3, the other will be 3 to 5. Okay, both are anti-parallel to each other. So these two are anti-parallel to each other. Understood? So one will be 5 to 3, the other one is 3 to 5. Both are anti-parallel. And complementary, we say, because adenine of one strand will always binds with thymine of the other strand. Guanine of one strand always binds with cytosine with the other strand. So anti-parallel as well as complementary. Clear? So that's how the DNA and RNA. So DNA both generates a replicate, provides the scope for slow changes, but DNA is a very stable, RNA is not stable. Why the RNA is not stable is it is having two reasons. Okay, so one is on the second carbon, it is having OH group, which is a very, very reactive group. which makes it less stable. So this is one reason. Second thing, in place of thymine, it has uracil, which is making it less stable. Okay, so these are the two reasons why the RNA is not chemically stable or structurally stable, but DNA is, because DNA is having the OH group. Sorry, RNA is having the OH group on the second carbon, which makes the RNA very reactive, and it's having the uracil in place of thymine, which also weakens the RNA. And both express the Mendelian characters. So apart from this, DNA and RNA both are same. So now quickly calculate this number and inbox me what is the answer. Come on. So you are given with the how many base pairs were present, and we know the distance between the two base pairs. So calculate what is the total length of this DNA and tell me the answer. Banu, are you there? She's not there. Okay. Mridula and Shrikar, tell me what is the answer. మృదులా చేస్తున్నావా?
చేస్తున్నా.
ఓకే. Yeah, Mridula, it's correct.
ఓకే.
ఎరా శ్రీకర్ నీకు వచ్చిన ఆన్సర్
Yes, sir. I'm sending you.
ఎంత వచ్చింది చెప్పా పర్లే.
The 1.56 in 10 raised to 6.
10 to the power of
6. I mean, if you want to reduce, then yeah.
10 to the 6 ఎలా వచ్చిందిరా?
So 1.56 into 10 raised to 6.
Okay, you didn't put this 10 power minus 9.
Yeah.
4.6 into 10 power 6 into 0.34 into 10 to the minus 9. So if you multiply 4.6 into 0.34, how much it is coming? Around
One-
like 1.6.
1.56.
That's 1.6.
Okay.
10 power 6, 10 power minus 9, it's a 10 power minus 3 meters. Because we are making in meters, right? Now if you see 10 power minus 3, that's nothing but millimeter. So 1.6 millimeters. Now see here, this 1.6 millimeter, okay, it is present in the 2 into 10 power minus 3 millimeters. That means the DNA is much more, many times bigger than the cell of the prokaryotes. But still it is able to pack inside it. And if you see the same with the eukaryotes, it will be around like 6.6 into 0.34. You will be getting 2.24 nanometers. Okay, so you see here, sorry, meters, not nanometers, 2.24 meters. And if you see the size of the nucleus, it is 0.000001. In this small size, 2.24 meters DNA as fitted. So how could this be possible? So there is a possibility because of a very, very complex involvement in the packaging. Okay, that involve a special type of proteins. They are called as the histones. So this complex packaging is happened because of a kind of proteins in the eukaryotes So in eukaryotes, so proteins are involved for packaging of this DNA. So those proteins, we call them as the histones. Histones. So what are the histones are present here? One is H2A, H2B, H3, and H4. Like that, there are two numbers were present, H2A, H2B. So these are all double copy were present. So H2A, H2A, H2B, H2B, H3, H3, H4, H4. Okay. So all this making a histone octamer, or you can say the nucleosome body, or you can say the nucleosome core. And there is another histone protein is there, H1. I'll tell you what is the use of this H1 later. Okay. Understood? So what is a histone octamer, or what is nucleosome core or nucleosome body?
Yes, sir.
There are total 8 histone proteins are there, plus 1, 8 plus 1. So here the 8 is H2A2, H2B2, H3s are 2, H4s are 2. So total they are making 8. Now this histone octamer, it is wrapped by the DNA. Okay, almost it was wrapped 1.7 turns. Okay, this 1.7 turns, it contains 200 base pairs of DNA. Okay, so you see here, this is the octamer. This octamer is just wrapped by the DNA, which is 1.7 tons, which is making 200 base pairs of DNA. So that structure, we call it as the nucleosome. Remember this. So the histone octamer plus DNA wrapping around it 1.7 turns, we call it as nucleosome. You can say DNA plus histone octamer is nothing but the nucleosome. Understood? And two nucleosomes, they come together, it will called as the chromatosome. Okay. And how does this nucleosome and the DNA are packaged? Because of the hydrogen bonding. Remember this. So there is a hydrogen bonding is present between the OH group of in amino acids of this histone peptide, or you can say the histone octamer, and there is a phosphodiester backbone is there, right, in the DNA. So there is a phosphodiester backbone. So this phosphodiester backbone of DNA and OH group in amino acids of histone, so they are making this hydrogen bonding. Remember this. So these two are making the hydrogen bondings. So that we call it as, I mean, that's how this wrap has happened. Okay. And to prevent this unwrapping, the H1 protein will act like a glue, which will seal it. Okay. So this may unwrap. To prevent that, we have the H1 protein. Is that clear? So see here, between the two nucleosomes, whatever the length of this DNA that is there, we call it as the linker DNA. So linker DNA is nothing but the DNA strand that is extending from one nucleosome to the other nucleosome in a chromatosome. Remember this. The small segment of the DNA that is connecting two nucleosomes. We call it as the linker DNA, or this is also called as interbead DNA. Okay, or you can say the linker DNA. Is that clear? Any doubts?
No, sir.
And whatever the H1 protein is there, it was present on this linker DNA and it will prevent the unwrapping of this nucleosome. Okay. So what is chromatosome? Chromatosome, it's nothing but the pair of nucleosome plus linker DNA. Together, we call it as chromatosome. Remember this. Now, like these group of nucleosomes on a linker DNAs, we call it as chromatin. Okay. And if you see these nucleosomes on this chromatin material, it will appear like the beds on thread. Okay, the beds on thread-like structure. So which is also known as the chromatin. Chromatin name came because it is a colored body when it is strained. So you add some color, it will absorb that color and it will look like that color. So that we call it as chromatin. And remember, the diameter of this chromatin is 10 nanometers, in case if some question comes on that. Understood. So just remember, it will give the string appearance under the electron microscope. On this string, there are beds were present, which are nucleosomes. So it's called as the beds on thread or beds on string. Both are same. Is that clear? Now this
Yes, sir.
long thread of chromatin, it was supercoiled, okay, with the help of another kind of proteins. Remember, for making this chromatin, we need a protein that's called histone. But for additional complex folding or supercoiling, we need another set of proteins. Those are called as the non-histone chromosomal proteins. Remember this. It is also said as the NHC proteins. The non-histone chromosomal proteins is called the NHC proteins. This will be required for uh, additional coiling of this DNA. So whatever the beds on string is there, now it was folding like this. It went on several foldings. Okay, so that's this one. And this structure, whatever this structure is there, it was further coiled like this. Okay, so you can see here. So super coiling has happened. That super coiling just folded in such a way like this. So which is giving a kind of chromatid structure. And there is a pair of chromatid structures which are joined at this. We call it as chromosome. Understood? So remember, this structure is happened because a lot of threads just folded like this. And it was given this kind of structure. We call it as chromosomes. And see here, when you observe these chromatin fibers under the microscope, they will give two color strands. One is the lightly strained, the other one is the dark strained. You can see here, the blackness, that is a dark strain, and there is a light color, that's a euchromatin. Remember the word euchromatin for the light strain and heterochromatin for dark strain. If you see this euchromatin and see this heterochromatin, in this euchromatin, there are 4 nucleosomes are there. In this heterochromatin, within the same area, there are 8 nucleosomes are there. So from that, you can say euchromatin, it is loosely packed. And if you see the heterochromatin, it is very densely packed region. Remember this. So euchromatin will stain light in color, stains lightly, or you can say light color. Whereas you see the heterochromatin stains dark color. And see here, if you want to create a RNA from the DNA, you cannot do that when the chromatin is so tightly packed. You can only do when the DNA is freely present. Even if you want to make another copy of DNA from the DNA itself, the replication, you need the packaging very loosely. Okay, so that's why this euchromatin is called transcriptionally active. Whereas the heterochromatin, we call it as transcriptionally inactive. Is that clear? So remember this, euchromatin loosely packed, heterochromatin densely packed. Euchromatin stains light, heterochromatin stains dark. Euchromatin transcriptionally active, heterochromatin transcriptionally inactive. So whatever the dark strain, it's heterochromatin. Whatever the light strain, it is a euchromatin. And remember this chromatin, it is a coloring object, so it can stain. Clear? Understood? So this is how a DNA packed inside the nucleus. Any doubts?
No, sir.
No, sir.